All right. Well, welcome everyone to our bi-weekly seminar. We are thrilled today to have Professor Keith Erickson from the University of Pittsburgh come and address the group with regard to physical activity contributions to brain health. Dr. Erickson currently is the director and principal investigator of the Brain Aging and Cognitive Health Laboratory at the University of Pittsburgh and the Center for Neural Basis of Cognition. Dr. Erickson studies the impact of physical activity and obesity on brain health across the lifespan. This research has resulted in approximately 250 published articles and 15 book chapters. His research has been funded by numerous awards and grants from the NIH, the Alzheimer's Association, and other organizations. He received a Distinguished Scientist Award from Murdoch University in Australia in 2018 and maintains a visiting professorship at the University of Granada in Spain. Dr. Erickson was a member of the 2018 Physical Activity Guidelines Advisory Committee and the chair of the Brain Health Subcommittee that was charged with developing the second edition of the Physical Activity Guidelines for Americans. His research has been featured in a long list of print, radio, and electronic media, including the New York Times, CNN, BBC News, NPR, Time, and the Wall Street Journal. Hopefully so. Without further delay, take it away, Dr. Erickson.
Great, thank you so much. That's a really nice introduction, and I'm really happy to be here and to present to all of you today. Of course, it's still several years into the pandemic, and I think we've in some ways gotten used to this, but I guess some ways maybe not. Of course, please let me know if you have any questions, and if you, in this part kind of environment, sometimes it's a little bit difficult to ask questions, but if you want to reach out to me after my presentation, if you find that easier or maybe more comfortable for you, please feel free to do that. I'm happy to communicate over email. Let me see if I can share my screen here. Good. So, most of my presentation today is going to be focused on brain health in older adults, although I'm going to touch a little bit on a few other populations that have shown some promising avenues, especially maybe how some of those prior studies or studies in other populations can help inform some of the results that we see in older individuals. First of all, we should start out thinking and talking a little bit about various perspectives on aging. And what I mean by that is that there's maybe some individuals that have shown a bit more optimism about the course of aging. Robert Frost once wrote that the afternoon knows what the morning never suspected. We often forget about the experience and wisdom that accompany aging, the experience of aging, and it's something that I think that we should always try to keep in mind. Others have, of course, been less optimistic. Mark Twain once wrote, "When I was younger, I could remember anything, whether it had happened or not. But my faculties are decaying now, and soon I shall be so I cannot remember any but the things that never happened. It is sad to go to pieces like this, but we all have to do it." What's really interesting to me about this sentence that Mark Twain wrote, "It is sad to go to pieces like this, but we all have to do it," is that we still talk about this. We still talk about whether we all have to go to pieces or not. The question here is cognitive change or cognitive decline or cognitive impairment an inevitable consequence of aging, or is it something that maybe we can slow down, we can prevent, we can delay? These are really big questions that we still talk a lot about today. And Mark Twain, I think, really picked up on this even more than 100 years ago now. In fact, when we start looking at data, of course, we see some very common patterns of decline across many domains of cognitive function. So this is data going back to 1994, Werner Schee's work, longitudinal work. And what we see is across numerous cognitive domains, some relative stability or even some improvements in some cognitive of these cognitive domains, and then some gradual decline after that, maybe hovering right around the age of 60 or so, maybe more or less. That's a big point of discussion as well, exactly when we should start or when we might expect to see changes happening in cognition. But changes happening in cognitive performance are usually preceded by a number of changes happening in the brain, including brain atrophy. And this is cross-sectional data from about 200 individuals between 18 and 80. And we measured the size of the dorsolateral prefrontal cortex. Here you see in the blue mesh representation. And what we see here is a gradual decline, actually fairly linear decline, starting at around the age of 30. Unfortunately for all of us, longitudinal studies have shown very similar patterns that the prefrontal cortex actually starts to show some deterioration right around the age of 30. We can look at other regions like the hippocampus. The hippocampus is located in the medial temporal lobe, here shown in orange. So it's sitting essentially right behind your ears in the middle portion of the brain. And here we see a slightly different function. Relative stability in the size of the structure until right around 50 years of age. And then we see gradual decline in the size of the structure after the age of 50. This structure deteriorates somewhere in cognitively normal older adults. It deteriorates somewhere between 1% and 2% per year. This structure is really critically involved in memory formation. In fact, deterioration of the structure is one of the more important predictors of conversion to Alzheimer's disease, where we see more widespread and significant impairments in memory function. When we look at individuals with Alzheimer's disease, the rate of decline or atrophy of the structure increases somewhere between maybe 3 to even 5% per year. The dorsolateral prefrontal cortex doesn't show that same level of atrophy, but it is one of the earlier regions to also to show age-related decline, as I discussed earlier. And so, and it's also critically involved in many higher order functions. So selective attention and executive function, working memory, these types of processes are generally supported, at least partly by the dorsolateral prefrontal cortex. Now, the average decline here is not really, it's not anything novel. Unfortunately, we see this across many studies. But what I want to point out here to you is the level of individual variability that we see in these scatter plots. So you can take this individual here who's about 70 years of age. They have the size of the prefrontal cortex equivalent to somebody actually much, much younger. This person, on the other hand, is about the same age and has the size of the prefrontal cortex equivalent to somebody much, much older. And we see similar types of individual variation in other structures. And so the question that emerges from some of these data is what factors explain this individual variation? Are these individuals that we see up here, have they just been genetically lucky to kind of show some prevention or some sparing of the tissue loss that seems to happen so frequently with aging? Or is it something that they've done throughout their lifespan, maybe, that has allowed them to age more successfully or to have some sparing of brain tissue even into late life? Of course, there's also the potential for methodological error. Of course, there's methodological error with any type of instrument, and so we have to also consider that. Nonetheless, across many, many studies, we see this type of individual variation that we want to try to provide some explanation for. And this is really where physical activity starts to emerge on the scene. And we can start to examine physical activity in relation to brain metrics or behavioral outcomes related to the brain, such as cognitive function, thinking, reasoning, memory function, and so on. But we can also think about the impact of physical activity and exercise on measures of mood, depression, anxiety. We could also think of perceptions of quality of life, which is also a very cognitive, measurement or cognitive aspect of well-being. Or sleep as a measure of healthy brain function. We can measure aspects of sleep quality and sleep duration, wake time after sleep onset, and so on, as measurements of brain function that could be affected by our various lifestyles, including physical activity and exercise. So when we look at the effects of exercise on the brain across the lifespan, and as we review this literature for the 2018 Physical Activity Guidelines Committee, we examine this really throughout the lifespan. And we can say quite conclusively that there's really no data on very, very early childhood. There's really not enough information about the impact of various movement patterns and activity patterns on brain development or cognitive or other aspects of cognitive development or brain development at that young age. So we don't really have much to say. In more school-age children, maybe around the age of 7 to 9 years old, 10 years old, we have a lot more data. And so I think we can be much more confident that about some of the implications and some of the conclusions about physical activity on brain health in this age range. However, in the more teenage adolescent years, we also have somewhat of a gap in knowledge. There's been significantly fewer studies in this age range, and the studies that have been done are usually cross-sectional correlational studies. And so there's really something of a gap. In midlife, we have a bit more information, especially with respect to how physical activity might influence aspects of mood, depression, anxiety, sleep outcomes, and so on. There's arguably less about the impact of physical activity and exercise interventions on cognitive outcomes or other maybe neuroimaging outcomes in midlife. The bulk of the data, the bulk of the evidence really comes in late adulthood when we start examining the impact of physical activity on neurological outcomes like Alzheimer's disease or dementia more broadly, as well as interventions that target individuals to examine whether we can modify cognitive function in this age range. So this is really where the bulk of the evidence comes from, and really where I'll be focusing a lot of my attention on today. However, it's important to point out that there's a few red X's on here, and it is important to keep in mind that the absence of evidence is not evidence of absence. So just because we lack data for a particular population or a particular age range doesn't mean that physical activity doesn't affect that age range, of course, or population. So it's just where there's gaps in knowledge that we need to provide. We need to have more evidence, more data in order for us to be more conclusive with some of our statements about the benefits of physical activity in that age range. So like I said, most of the data is really coming in relation to the benefits of physical activity in late adulthood. Before I jump into some of the data, I think it's good to maybe take a step back and remind ourselves of some of the terminology and design, experimental designs and measurements that are often used in this literature. So starting up on top, we have more experimental designs, which are really interventions, randomized clinical trials in which individuals who volunteer for participation in the study are randomized to maybe one of two conditions, two groups, either a control group or to a treatment group that receives some amount of physical activity or exercise that is well controlled. Typically, we try to keep this as well controlled as possible. But things that we might consider as in the design of our interventions are things like the intensity of the intervention, the time or duration, minutes per bout or per week, the volume of activity per week, the frequency of the activity, the intervention length for 12 weeks or 6 months or 52 weeks, 1 year, even longer than 1 year, as well as the mode of activity, whether it's walking or another type of mode like dancing or resistance training. And so this is much more rigorous type of design, and you can tell a lot more from this type of design as to the extent to which cognitive function or any type of outcome that we're looking at is potentially modifiable by engaging in exercise. And so from that perspective, we can think of this as a way of potentially, as a therapeutic approach. We could use physical activity as a way of treating a particular condition or as a way of preventing further decline as well. In contrast, we have observational studies that consist of more observational longitudinal studies in which we measure, for example, physical activity patterns early in life and then follow people over years or even decades to examine whether engaging in activity earlier in the lifespan alters risk associated with some disease outcome later in life, or cross-sectional or correlational studies. And quite often, cross-sectional correlational studies, we can't obviously make causal conclusions, but they're often very informative in terms of providing some evidence pointing us to a certain direction that can then give us some insight or some guidance regarding how interventions might be designed. And from this, we can measure things like cardiorespiratory fitness levels. We might engage individuals with a self-reported, more physical activity questionnaire, or use some device monitoring, accelerometry or actigraphy types of devices to try to capture information about activity patterns. We also want to, before I jump into the data, we might also want to think a little bit about how physical activity might be working. And I'm going to talk a little bit about mechanisms in a little bit more depth a little later in my talk, but here you can see that various cardiovascular risks, metabolic risks, diabetes, early life adversities, the presence of early life adversity early in the lifespan might influence brain development or brain health throughout the lifespan and influence brain outcomes even late in life and even possibly risk for Alzheimer's disease. And many of the possible pathways that are examined or have been speculated as playing an important role in some of these cardiovascular metabolic risks and early life adversities are thought to influence brain health in late life. It turns out that physical activity might actually be playing a role on some of these same molecular pathways. And so it's possible that, that physical activity is working through some of these ways to influence or to counteract some of the negative influences of cardiovascular and cardiometabolic risks throughout the lifespan. We look at specifically certain disease outcomes, and again, we can think about influencing risk for certain diseases like Alzheimer's disease. We can examine various meta-analyses, and this was from a meta-analysis in 2015, and in particular, this meta-analysis summarized observational longitudinal studies, so examining whether greater amounts of physical activity a few years earlier in the lifespan was associated with increased risk of developing Alzheimer's disease specifically as a form of dementia, Alzheimer's disease specifically. And what you can see is that from this meta-analysis, greater amounts of physical activity was associated with about a 50% reduced risk of developing Alzheimer's disease. So this is a very important finding, of course, but as some of you might be realizing, there's a lot of challenges with interpreting some of these data. In fact, there might be some reverse causality happening, right? So maybe as the brain starts to deteriorate, movement patterns, balance issues, and other things influence individuals' participation in physical activity. And so maybe physical activity or the reduction in physical activity maybe a consequence of a deteriorating brain and a sign of neuropathology rather than a causal risk factor, causal factor influencing risk for Alzheimer's disease. So this is something that we can't really glean from these types of studies. But yet, nonetheless, they are very interesting and very informative, and still is clearly a risk factor given these particular data. In contrast, we can look at other populations and look at results, meta-analysis results from meta-analyses examining the impact of interventions, randomized clinical trials on various outcomes. This was a meta-analysis published by my graduate student, Lauren Oberlin, in 2017, who's now a postdoc up at Cornell. And what we did was examined trial that randomized individuals who've experienced stroke. And they randomized these individuals to either receive a physical activity intervention or a control group. And generally, the control group is often like a usual care type of control group. And they examined changes or improvements in cognitive function resulting from the intervention. And what you can see here is across these studies, we see some consistent patterns such that exercise, participating in an exercise intervention, seems to improve cognitive function across these studies. And this effect size was about 0.3. So the randomized clinical trials show that, that exercise can improve cognitive function in stroke survivors with a smallish effect size. And I'm pointing out this effect size of 0.3 because actually when you look at meta-analyses across most interventions, most meta-analyses of intervention studies, whether we're looking at children, midlife adults, older adults, Alzheimer's disease patients, stroke patients, and so on, we see very similar effect sizes come up. They're all hovering right around 0.3. Here's another example. Plenty of studies have examined the potential for physical activity to actually influence cognitive function in people with schizophrenia. And so here too, these are randomized clinical trials. Individuals with schizophrenia are randomized to either an exercise condition or a controlled condition. And here too, what you see is across these studies, exercise seems to improve cognitive performance with an effect size of 0.33. Again, very similar effect sizes popping up across these meta-analyses. When we look at cognitively normal older individuals, and by cognitively normal, I'm generally referring to these people have not been diagnosed with dementia, but they're all older individuals, typically 60, 65 years of age, up to most interventions around up to about 80 or 85 years of age. And this description, this meta-analysis actually is described in a slightly different, or is being illustrated in a slightly different pattern, but this is all from randomized clinical trials. And this particular meta-analysis divided the cognitive domains into 4 different groupings. Now, there's 2 real take-home messages from this particular data, from this particular meta-analysis. One is that you can see that the effects of exercise for improving cognitive performance seem to be fairly broad. That is, most cognitive domains showed some improvements with engaging in exercise relative to the control group. However, even despite some breadth of the effects, there's also some specificity. You see that this bar, the executive functioning bar, seems to be higher or is higher than some of the other bars. And so this suggests to us that there's some specificity to the effects as well, that executive functions seem to be affected or enhanced more so than other cognitive domains. And so this gives us maybe some insight or maybe some clues as to what brain regions might be more affected by exercise than other brain regions. Certainly, maybe when you look across all of these domains, maybe the entire brain is affected in some capacity by engaging in exercise, but maybe some brain areas are affected more than others. Why that might be is actually a very important question. Memory tasks, there weren't really too many memory tasks when this meta-analysis was published in 2003, but they were generally grouped under these controlled processes. And so you can see that there's still some effects here. And I'm going to come back to that because we've got a much better sense now of what's happening in memory domains specifically. Nonetheless, this gives us some idea of what might be happening in the brain. So we approach this saying, let's hypothesize some of the brain regions that might be affected. And here we specifically focused on the hippocampus. The hippocampus is, as I mentioned earlier, deteriorates with age, is a risk. The deterioration is a risk factor for Alzheimer's disease. It's closely linked with memory function. And decades of work in animals, in rodent studies specifically, have very clearly demonstrated that exercise affects this structure, the integrity and functioning of this structure. So let's take a deeper dive into the hippocampus and see if we find something there. The other brain region, given the previous results that I just described, was the prefrontal cortex, which I described earlier as involved in supporting executive function. So maybe these areas might be some of the key areas involved in in exercise interventions or being affected by exercise interventions. So that's what we started hypothesizing, what we started to take an initial look at. Now, let's start with the hippocampus. And in this review paper that my group just published, Sarah Agajayan and Alina Lesnovskaya were the two lead authors on this paper. You can see here in this diagram that you can divide some of the hippocampus neuroimaging markers essentially along two axes. You can think of them more in terms of morphological data, so the volume and structure of the hippocampus, or the white matter, the white matter paths and tracks that are linking the hippocampus to other brain regions, like the fornix, for example. We can also think about connectivity or the functioning of the hippocampus, or cerebral perfusion of the hippocampus. We can look within the hippocampus, or we can look at how the hippocampus is communicating either structurally or functionally to other brain regions. So I'm going to talk a little bit about some of these results. So back in 2009, which of course doesn't seem like that long ago, but it was over a decade ago, we came out with one of the earlier studies, the initial studies examining some of these associations. And here again, as I mentioned, this is a cross-sectional study, and cross-sectional study that has all the limitations of any cross-sectional or correlational study, but you never want to invest a lot of time and resources into a really massive randomized clinical trial with neuroimaging data before and after the trial. You don't want to invest a lot of that unless you have some good sense of what's happening in a cross-sectional or correlational perspective. And that could also really help guide the development of a grant proposal or hypotheses. So we examined 165 adults between 59 and 81 years of age, free of dementia, and we put them onto a treadmill, gave them measured cardiorespiratory fitness, aerobic capacity, aerobic fitness, and we put them into an MRI machine, and we used an automated segmentation algorithm to identify the location and shape of the hippocampus, which you see here. And what we found was quite striking. Cardiorespiratory fitness levels are down here, and volume of the hippocampus is here. What you see is a very striking association. Individuals that are higher fit tend to have larger hippocampal volumes than their lesser fit peers. Now, remember, this is at an age in which there's typically deterioration of this structure. Also, deterioration is predictive of conversion to dementia or to Alzheimer's disease specifically. So typically, larger volumes of the hippocampus at this age range is a very important thing. So this is quite provocative. Now, there's a few things with this. This association remains significant even after controlling for variation associated with age, sex, years of education, and intracranial volume. So it's not just that we're capturing all males who have larger heads up here, or it's not that we're just capturing younger individuals up here, and these are tend to be older people. So even when we control for some of these variables that I just mentioned, this effect remains significant. The other thing that's really striking is that this study now, since 2009, this particular association has been replicated numerous times in the literature across different age ranges. Even in children, we see this association. Children, adolescents, midlife adults, older adults, we see this quite frequently. So there is something going on here, between cardiorespiratory fitness and hippocampal volume. Stated in another way, if you remember back to one of my initial slides, I said that there's significant individual variation in the size of the hippocampus and hippocampal decline. And it appears that cardiorespiratory fitness levels are at least one measurement that explains some of that individual variability. But that's just cross-sectional data, right? So it's provocative, but it doesn't tell us whether modifying cardiorespiratory fitness would actually modify the size of the hippocampus. To do that, we need to use more of a randomized clinical trial. And so we did that. This is the design of our typical randomized trial. We bring in older, sedentary, more inactive adults into the study. We do a variety of baseline assessments, fitness testing, MRI, cognitive testing, and we usually take blood for various biomarkers. We randomize them to either one of two groups, typically, either to a brisk walking group or to a stretching and toning control group. So the similarities and differences between these groups are really important to understand. Both groups are receiving physical activity around a track. So even stretching and toning, stretching is a form of physical activity, but we make sure that they're doing it at a light intensity. Both groups are receiving some laboratory-based treatments. In other words, it's not just an unsupervised intervention, it's happening under very supervised conditions. Both groups are coming into the lab for the same amount of time. So they get the same amount of instruction from our exercise trainers and exercise physiologists, and they get the same amount of social interaction. So the main differences here are the intensity and the type, right? Walking versus stretching, and then brisk walking, we measure their heart rate, make sure that they're really working at a moderate intensity level during the course of the intervention. We typically do this for 6 months or 1 year, and then we do all of the follow-up assessments, the same assessments that we did at baseline. So in 2011, we published a big study with 120 adults, published in PNAS, and we had slightly more women in the walking exercise group here, but all the effects that I'm going to show remain significant even when controlling for sex differences. You can see attendance rates were about the same in both groups, right around 80%, which is quite good. And we reliably improved fitness levels, cardiorespiratory fitness levels in the walking exercise group. There was not a reliable change in cardiorespiratory fitness for the stretching control group. When we looked at we started looking at the morphology, the size of 3 different brain areas, the thalamus, the caudate nucleus, and the hippocampus. I'm going to focus right away here on the hippocampus, because this was really the only significant finding from these other brain regions, and kind of goes in line with what I was just talking about before with the cross-sectional data from the hippocampus. And so you can see here a slight decline in the size of the hippocampus over a 1-year period in the control group, but a slight increase of about 1.5 or 2% in the both left and right hippocampus for those individuals engaging in exercise. So this particular effect demonstrates a remarkable degree of plasticity in these older individuals. Something that has been a major question, right, for many decades is how plastic is the older adult brain? Well, these data seem to indicate that at least the hippocampus retains some of its natural capacity for plasticity. And it also indicates that engaging in 12 months of moderate intensity exercise might be capable of taking advantage of that plasticity and modifying the hippocampus in a positive direction. And this is, of course, really important for in the context of Alzheimer's disease and risk for dementia. We might be, in other words, exercise might be influencing risk of dementia by its effect on the hippocampus. We've now done, this was done in 2011, my study, but we just recently published a meta-analysis in 2021 across all studies that have been interventions that have examined the effects of exercise on hippocampal volume. You see a significant effect, but the effect size is actually quite small. Now, what's not really shown here is that this effect size was actually moderated by the age of the sample. So this is across everyone, not just older adults, but there's some studies here in children, some studies here in other populations, including schizophrenia, for example. And generally, the effect sizes here, the effect size actually increases with increasing age. So we know that the effect size is larger for older adults, which probably makes a lot of sense because that's when the structure is really deteriorating. Remember that typically a lot of deterioration in the structure until about the age of 50. So we start focusing on individuals over the age of 50. That's when we might start really detecting some of the positive effects of exercise on the structure. We next asked whether improvements in fitness were associated with changes in volume. So what we did here, what you see in this particular scatter plot is change in hippocampal volume versus change in VO2 max, percent change. And you see very clear association. So this is indicating to us that some changes in cardiorespiratory fitness might be a necessary prerequisite for seeing changes in hippocampal volume. However, that is still speculation because these are correlations. We still have not really determined whether this is essentially a mediator for some of the changes. But nonetheless, this is a very intriguing and striking correlation. When we look at now the literature across the hippocampus, still again focusing in on the hippocampus, this was recently published by a group in this review paper. But what you can see here is that the majority of the studies certainly have been in cross-sectional studies looking at cardiorespiratory fitness across hippocampal volume. Even in white matter, we see most of the effects or most of the published studies coming up with cross-sectional studies. So there's considerably fewer intervention studies. That's a major problem. We need more intervention studies, especially at this point in time where there's enough cross-sectional data for us to be confident that there's something going on. We just need to have well-designed interventions. You can see hippocampal volume. There's certainly more interventions, as we just discussed, than there are in, for example, in white matter. I'm going to come back to the white matter effects here in a little bit because I'm a little bit more skeptical of some of the white matter patterns. But you also see that there's a gap in knowledge here with respect to other measures of connectivity or perfusion in the hippocampus. There's just not a whole lot published right now in some of these areas. And we're trying to fill some of these gaps with some of our current stems now. Another question that you might be asking is, well, who cares about the volume of the hippocampus if it doesn't really translate to improvements in memory function? So is there evidence that exercise actually improves episodic memory function? Episodic memory being really thought to be a cognitive domain very reliant on the hippocampus. Well, I can tell you that there's been a lot of debates over a many years. And so we tried to resolve this issue by conducting a meta-analysis. And this is a really nice paper that I'm very proud of my graduate student, Sarah Gajayan, for producing. This is in Press. And this is a meta-analysis of randomized clinical trials in older adults, examining whether the participation in the intervention improved episodic memory performance. Okay, so you can see that now, as of 2021 or so, there's been quite a few studies that have been published in this area. And part of it is that there's a lot of work going on in the hippocampus. So what's happening in episodic memory? Well, one thing that you see here is that there's a significant effect of exercise on improvements in episodic memory performance with an effect size of 0.28. So again, hovering right around that 0.3 that we're seeing across all of these other meta-analyses. We also did a leave one out cross-validation so that we could, kind of control for maybe some of these outliers, and the effect was still significant. Even controlling for this one or removing this one actually increased the effect size a bit. But the other thing that I want to point out here is that when you look across these individual studies, most of them were not significant by themselves. It was only in the context of a meta-analysis where we could really pool all these studies, pool all the data, and really detect some effect. So the next thing that we did was we did a power analysis based on this meta-analysis. And what we found was that with an effect size of 0.28, we would require about 202 people per group to have a sufficiently powered study. Now, what's really important is that none of these studies actually had 202 people per group. So almost all of these studies that have been done in the past have actually been insufficiently powered. And that's actually something that I think is contributing to some of the muddiness in the literature and some of the heterogeneity that we're seeing and sometimes reading about. Now, the hippocampus isn't the only region. As I mentioned, the prefrontal cortex seems also to be affected. This was a study, an intervention back in 2006 that we found, that we published rather, and we found results, improvements, increases in various regions of the prefrontal cortex. But one thing that I want to point out here with these particular images is that it's not that the whole brain is just getting larger over the course of the intervention. There's somewhat regionally specific effects, not just the hippocampus, but regionally specific effects in the prefrontal cortex. And when we look at cross-sectional data, we see similar patterns. Cross-sectional data showing that whether we look at physical activity or cardiorespiratory fitness, that individuals engaging in greater physical activity or higher levels of fitness tend to have greater gray matter volume in all of these colored areas. So we're seeing a lot of consistency. With that measurement, the morphology, the volume structures, or the volume of the structures that we're getting, so volumetric studies. When we turn our attention to white matter, the patterns change a little bit, as I alluded to a few minutes ago. So our numbers that even just a few years ago, our white matter measures weren't nearly as of high quality as they are now. And so we can now have much higher, finer resolution of our imaging techniques. We can better disentangle some of the crossing fibers and determine crossing fibers a little bit more clearly. And so we can use these techniques as well to try to examine how exercise might be altering white matter connectivity or white matter tracts and tract integrity, either cross-sectionally or as a function of an intervention or observational study. Generally, if we start out just cross-sectionally, there seems to be a lot of consistency in the patterns, especially when we, cardiorespiratory fitness, relational matter integrity. Here's a study again by my graduate student, Lauren Oberlin, across 2 separate samples. We found very clear associations. Those people that were higher fit tended to have higher white matter integrity throughout many regions of the brain, actually, including these white matter tracts. And so, especially in the prefrontal cortex, you see. So this is very clear. And it's not just this study, there's been numerous other studies finding these associations. Where it's less clear is whether interventions actually modify white matter. I think I'm skeptical of that at this point. And it's primarily because I think when you look at animal literature, it's not that white matter isn't plastic. I think that there is good evidence that it is, but I also think that it might be less plastic than some gray matter regions. And so it might be that gray matter regions are more plastic, more amenable to more quick or rapid changes occurring as a function of maybe a 6-month intervention. White matter, I think, is probably a bit more protracted, and cardiorespiratory fitness or cross-sectional associations might be picking up maybe a lifetime of healthy behaviors that we're just not seeing when we get people who have been inactive most of their life and just get active for 6 months or 12 months. But like I said, the jury's still out. We can also measure measures of functional connectivity. And here too, there's a number of ways of examining functional connectivity, either during a task or at rest. And a number of studies from our group and others have been finding that exercise training actually increases connectivity. Now, what's really interesting is that you might ask, well, which brain areas show increased connectivity with engaging in exercise? Well, you see here the hippocampus and part of the prefrontal cortex and medial wall, the same regions that I was just mentioning that seem to be affected in terms of morphology. Here too, in this intervention, we see the hippocampus and we see the prefrontal cortex. And so numerous studies keep coming up, whether we're looking at activation, whether we're looking at resting state measures or morphology, we see prefrontal cortex and hippocampus. And similarly, cross-sectional studies, just as I mentioned earlier with morphology, cross-sectional studies have been showing very similar patterns. That those individuals with higher fitness levels or those individuals engaging in greater amounts of physical activity tend to have greater connectivity or different patterns of connectivity. For an example, during a task, sometimes less activity can actually be related to better performance. You might ask also about cerebral blood flow, and we have been measuring cerebral blood flow, and this was a study published. We've been publishing from a randomized clinical trial published in 2020 and 2021, in which we had people randomly assigned to either a dietary condition, a dietary plus moderate intensity exercise, or a dietary plus high amounts of exercise. So this group was getting closer to about 150 minutes of exercise, and this group was getting closer to about 250 minutes. And one thing you see is that we see significant weight change, a decrease in weight across all three groups. Now, what was really striking was what we took cerebral blood flow measurements using arterial spin labeling techniques in MRI, and we looked at before and after the intervention, and we saw significant increases in cerebral blood flow in all of these red areas across all three groups. When we broke it down a little bit more, we saw that there's some specificity to these effects. So in the green areas, you see that the green areas were showing effects related to the diet-only condition, really not as much to exercise exposure. Whereas in the blue areas, you see that the effects were a bit more specific to the exercise conditions. And so why exactly we're getting this specificity? Why isn't it just an overall effect throughout more regions of the brain? This is still an open and unanswered question that we can speculate about, that we are speculating about. So you might be asking a little bit about mechanisms, and I'd like to talk a little bit about mechanisms or speculate a little bit about mechanisms before we finish up the presentation here. So we can talk about mechanisms at a few different levels: cellular and molecular, structural and functional, or behavioral, socio-emotional. And these are all three different levels that are not mutually exclusive from one another, but it's important to be thinking about this, and because a lot of us automatically think at more the reductionist level. How is this happening? What are the molecular changes happening in the brain that's giving rise to these changes? And Carl Kottman and many others have put models together talking about various molecular changes that might be happening. When we look across, however, when we look across various molecular pathways, we see that there's a lot of things that are happening in the brain at more of the cellular and molecular level. Changes in metabolic factors or insulin glucose signaling, cerebral vascular changes. There's epigenetic changes that we know happening at changes at methylation patterns at the genetic level, changes in neurotransmitters and so on. But we might also ask, to what extent do these structural and functional brain changes mediate cognitive benefits or mood changes or anything like that? But we might also ask, maybe changes in sleep behaviors or mood are actually mediating some of the cognitive improvements. So in other words, maybe exercise is a great way to get people to sleep better, and the improvements in sleep are allowing people to think better and remember things better. And if that's the case, then sleep would be acting as a mediator for the effects of exercise on cognition. I refer to this as the sledgehammer effect. In other words, exercise is not necessarily a precise approach. It's a sledgehammer to the body, we are manipulating and changing whole areas of molecular and cellular pathways, behavioral measures, and brain aspects. And that's one of the benefits, but it also makes it one of the challenges from a scientific perspective, tracking down the precise mechanisms at play. But when we look at animal studies, we see that exercise influences neurogenesis, the production and proliferation of new neurons, especially in the hippocampus. We also have some links to molecular factors. So brain-derived neurotrophic factor, BDNF, is a critical molecule involved in many different conditions and diseases of the brain. And back in 1995, and since then, many researchers have found that exercise increases the production and secretion of BDNF in the hippocampus and other regions. We studied brain-derived neurotrophic factor changes in my study published in 2011, and we found that individuals that showed greater changes in the hippocampus also showed greater increases in BDNF levels over the course of the 12-month period. Again, this is just a correlation, but it is very consistent with the animal literature on this topic, and so is nicely bridging some of these studies from animal rodent studies to humans. We've also been asking, maybe amyloid plays an important role in some of these associations, especially risk for Alzheimer's disease. And this was a study from the cardiovascular health study, a longitudinal observational study. So keep that in mind, 149 individuals. And we had physical activity measured at baseline, cognitive impairment measured 13 years later. And we have amyloid levels measured in this 9 years after measurement of physical activity. And what we found was that physical activity, greater amounts of physical activity, was associated with reduced amyloid levels 9 years later, but it was also significant statistical mediation such that reduced amyloid was associated with a reduction in risk of cognitive impairment several years after that, suggesting that changes in amyloid or a reduction in the accumulation of amyloid might be also acting as an important mediator. Now, we can also question about moderators. And moderators here you can think about as going out for a run is really beneficial, but if you stop at a bar on your way home and have a burger, beer, and fries, then you might be mitigating some of that benefit of going for a run. However, if you engage in an intellectually active environment or stimulating environment, that maybe the stimulation, the cognitive stimulation might be actually augmenting some of the benefits. So you can think about moderators from a few perspectives. A good example here is sex differences, because a lot of studies find benefits benefits of exercise are greater for women than men. But there have been a handful of studies finding greater effects for men as well. So we're still really trying to understand the reason for some of the sex differences, but they do seem to be there. Another example might be APOE genotype, which APOE genotype is, of course, the genetic risk factor for Alzheimer's disease. And this was a study published by Denise Head and colleagues in 2012. And it's a really striking because what they're measuring here is mean cortical binding potential of amyloid using PET technology. So higher bar means greater amount of amyloid. And here you can see these are people engaging in low amounts of activity versus high amounts of activity. And these are people that are E4 negative. They're not carrying the APOE4 allele for greater risk of Alzheimer's disease. And these people are carrying it. And so what you see here is that the bar that jumps off the screen here are these individuals right here getting both low amounts of activity and are a genetic risk for Alzheimer's disease. They have much higher amyloid levels than the other groups. But what's really important to me actually is not this group, but this group right here, because it seems as if engaging in higher amounts of physical activity is mitigating some of the effect of the E4 allele on the accumulation of amyloid. You can see that this group has the same amount of amyloid as these other two groups here who are E4 negative. So this is really striking and again shows a possible moderating effect of physical activity on a genetic risk for Alzheimer's disease. Unfortunately, most of these studies have been cross-sectional, so we still have a lot of work to do. We just don't have sample sizes large enough to really test this in interventions. And so we've come a long way looking at aerobic exercise, various growth factor cascades neuroprotection, neuroplasticity factors, and the cognitive and behavioral sequelae of brain function and plasticity. And this has given rise to various publications and policies prescribing physical activity for specifically influencing brain health throughout the lifespan. So we have many unanswered questions. How long are the effects of exercise retained? How long do they persist? We don't really have a good sense of that. What types of exercise are most effective? We still have a very poor understanding of the impact of various modes of exercise. What are the dose response effects? Still a poor understanding of that. And what do the volumetric differences really reflect on a cellular level? We're still trying to answer many of these questions, and many ongoing interventions by my group and others are hoping to address many of these ongoing and unanswered questions. For example, IGNITE is a phase 3 RCT in cognitively normal adults, 639 participants in a 12-month supervised intervention with a dose response manipulation. So we're hoping to address a whole variety of different questions, and we're collecting a very large battery of various measurements and outcomes in this particular study. So in conclusion here, we have many choices to make, healthy choices, dietary and physical activity choices, and these clearly impact brain health outcomes throughout our lifespan. Exercise has widespread effects on the brain. It's very clear now that we can be very conclusive about that. Moderate intensity exercise several days a week is sufficient for improving brain health, but that being said, we have a very poor understanding of the dose necessary. Starting to exercise in late life is not futile. Even those who are sedentary can improve function. But that being said, starting earlier in life is certainly probably better. Exercise may have long-term health consequences for many different diseases of the brain. And more research is now starting to examine the impact on brain outcomes across numerous populations. So this is my group, which I'd like to thank very much for all of their supports and contributions to all the studies and funding and collaborators. So thank you very much.
Okay, that was awesome, Kirk. That was fantastic. 2 weeks ago, we talked about, behavioral modifiable risk factors, one of them being the building and maintaining of cognitive reserve, right? And I use the analogy of the way we talk about skeletal health, whereas you accrue as much as you can in early life, and then in adult life, middle age, you do everything you can to maintain it. And then as you get older, you do everything to attenuate the slope of decline.
Yeah.
Right. So that's how we do bone health, right? Would you say a similar thing about the value of physical activity to brain health starting early in life?
This is a very, very good question. I've got my answer for you is yes, with some qualification to that. And what I mean by that is that cognitive reserve is usually in the context of dementia is classically discussed in terms of maintaining function in the presence of pathology. And so individuals that have some neuropathology, why do some of them still function, maintain some degree of decent cognitive function? And so that is, I think that is the classic kind of way that we often think about cognitive reserve. And in this case, physical activity has been talked about as one way of building reserve in order for somebody to continue or maintain function, even in the face of having some pathology. And I think that that's absolutely true. However, we can also think about physical activity, especially a lifetime of physical activity, as possibly a way of influencing the brain in such a way that the neuropathology doesn't show up in the first place, or doesn't build up at the same rate in the first place. Right. And so therefore, it's even a slightly different way of thinking about the brain, or thinking about cognitive reserve. that the cognitive reserve is actually happening through a lifetime of physical activity in a way that is not just building the reserve, but providing some protection against the accumulation of the pathology in the first place.